So with no further ado, I'm going to turn it over to Dr. Stover.
Thank you very much, Wendy. And thank you very much to Sylvia and the organizers of this session. I want to start out just talking a little bit about consumers and an experience I had a couple months ago where I missed a flight and I had to get somewhere, so they sent an Uber driver. And the woman was quite chatty at about 1 o'clock in the morning and told me about her 2 kids, told me about her husband who was an NFL player who was injured, and was no longer going to be able to play football. And she had started a career as a personal training coach. And that she had amassed a huge clientele in a big city, all these clients. And then when she learned that, I was in the nutrition area, she says, oh, well, for the people, my clientele, who are mostly obese, she said, nutrition is the most important guidance I give them. And I use the blood type diet. And I match their blood type to specific dietary patterns, and of course it's based on Peter Damber's book. And she said that's the single most important thing that improves their health is following the blood type diet. So I think that's where a lot of the consumers are. We can talk amongst ourselves about all of this, but it's actually blood type. But at any rate, these are my disclosures right here. All of my money is from research monies from NIH. I'm sorry. Is from NIH, and I do consult. So first starting out, I love this issue of Nature a few years ago, 2010, asking the question, can science feed the 9 billion people that'll be on this planet in 2050? And then you have this article here on diabetes. And the question is not if we can feed that many people, evidence is we can, but can we feed them in a way that keeps diabetes off the cover of Nature? And of course, this is because we have the unprecedented capacity, really, to formulate our food supply any way we want at this time. And the question really is, what are our expectations of the food supply? That is, when we think about setting dietary and nutrient guidance and recommendations, what are the outcomes we really want to achieve and what is achievable? And of course, most recently, the federal government spoke to this when it asked the National Academies to assemble a committee and develop a consensus report on developing dietary reference intakes based on chronic disease endpoints. That is, how can we give guidance of how people should eat based on chronic disease endpoints? So it is about connecting food and both disease prevention and health. And just these are my thoughts on that report. And number one, when we think about chronic disease and its connection with the food supply, few chronic diseases are affected by a single nutrient or a single pathway. So therefore, when we think about the relationship between food and chronic disease, we really have to consider systems networks over individual pathways. All of the pathways, how they converge, how they interact, how all the nutrients interact. That means when we think about biomarkers, we can't think about single nutrient biomarkers, for which there are very few, but we have to think about integrative biomarkers that are the endpoints of systems where pathways and nutrients interact that then directly bridge to chronic disease. Of course, the report calls for a range of DRI values in lieu of point estimates, which makes sense when you consider networks. And the biggest risk you have for chronic disease is to age. And so therefore, we really have to understand biomarkers of aging in the context of biomarkers of nutrition and how those two interact. And of course, the other recommendation that came out of there was that the use of grade for the standard of evidence. This is a field that's driven primarily by observational data. Most of that observational data will not grade at a level you need to set chronic disease endpoints. We also know that about half the population in the US, adult population, is under care with a physician for some sort of a chronic disease. So therefore, potentially, half the population, the DRIs, which are meant for healthy people, they may not directly apply. And because of that, now the National Academies is setting up another group to examine, I'll get this right here, to examine nutritional requirements in disease states. And this workshop is going to be held in 2018. So when we think about nutrigenomics, we really have to think about the totality of nutritional requirements, what they mean, and that is to classify and evaluate human nutrient needs across that span of health through disease prevention to disease management. And currently, right now, the DRIs are really focused on the nutritional needs of healthy individuals and the prevention, primary and secondary, of diseases. And we have these sorts of markers at our disposal to try to understand what those needs are. But increasingly now, we're in this space now of trying to understand the connection between food and disease trajectory, as well as disease management. And this means we have to have other types of considerations. We have to think about tissue-specific nutrient status. That is often chronic disease from its very early stages to late when it's fully manifest can exhibit tissue-specific effects that really may not relate to whole body effects. And so we need to think about individual tissues. We need to think about restoration of function. And we heard Dr. Morris earlier talk about the role of conditionally essential amino acids in certain disease states. And we have to think about tissue regeneration. That is, what are the unique nutritional needs of stem cells as they go to repair damaged tissue? And so where the dietary reference intakes really relate to the food supply, disease management, we begin to think about these distinctive nutritional requirements, medical foods, which provide nutrients that may not be accessible in the quantity or quality one needs from the food supply. So when we think about dietary requirements, we really have to think about them, as we've heard today, as complex traits. That is, all of these physiological processes inform what nutrient needs are, the interaction of all of these processes and many more. And of course, this has been recognized for many years now. This is 2013 when the American Society for Nutrition put forth their research agenda. And when you think about nutrient requirements as complex traits, then you expect to see responders or non-responders or a gradient of response. And therefore, one of the top priorities identified by the American Society for Nutrition was to understand the variability in response to diet and food among individuals. And that's what we're talking about today. And when the National Nutrition Research Roadmap was released by the federal government earlier this year, it also focused on that connection between nutrition and disease prevention, as well as understanding individual difference in nutritional status. So in fact, this has been an area of intense interest, one for which, though, is not completely related to, as we know, our genetic makeup. And when we think about, the effect of genetics on nutrition, we have to remember that when the Human Genome Project was completed, the purpose was not only to inventory all the genes and all of the variations within those genes among humans, but to really assemble all of these components of physiology, all of these proteins and interacting molecules into circuits, into networks, and to understand network behavior, and not only what the effect of a single gene variant is on a protein, but how all of these variations combine within networks to change or affect a system readout. And then once you understand that variation, how we can manipulate the inputs, whether it be a drug or a nutrient, based on that variation to optimize the function of that network. So when we think about then the impact of genetics and why that does affect diet, this is really rooted in evolutionary biology. And that is, this was a cover from 2002 of Scientific America that talked about food for thought, dietary change as a driving force in human evolution. And that is, we know that the molecular basis of the evolution of species is the evolution of genes, how genes change over time. And not all genes evolve or change at the same rate. Genes that evolve or change quickly are those that enable adaptation to a local environment. And so when you look, one of the most powerful selective pressures in genome evolution has been diet. And I'll speak to that in a minute. But historically, the presence of dietary components has affected our primary sequence, enabled genome program, and the regulatory effect of nutrients on gene expression. We also know now, with the identification of the Denisovan archaic humans, that in fact Denisovan DNA also became integrated into the modern human genome. And through these studies, we also understand now that there was probably at least a third potential unknown hominin that also contributed DNA into the current human genome. Why is this important? Well, it's important now because we understand now what the Denisovin and the Neanderthal genes are that have now integrated into the human genome, and how these have affected human traits, including susceptibility to certain diseases, our immune response, skin color, how we deal with UV light, etc. So this is one source of variation within humans that we know varies across the planet. And again, why this is important is because people will know this. Consumers love to do these hereditary tests. And this is off the internet, but this is from 23andMe, where you can not only find out your ancestry, but you can find out what your percentage of Neanderthal genes is, Denisovans, et cetera, et cetera. But with that comes a lot of information about your potential health status or health effects long term. We also know that within the genome, that variation within the genome has happened through mutation events that have then expanded within the population. And they've expanded either through selection or through drift. And just to show you how that occurs here, I think you're all aware of this, but if you have variation at a particular haplotype in the human genome, and you have a new mutation that arises, and that mutation enables some sort of a selective advantage for you to propagate your genes, you will wipe out pre-existing variation. And computers can pick this up. So a computer can identify a particular region of the genome or a particular gene that has conferred some sort of an advantage, displaces removes pre-existing variation, and then begins to form other variants over time once you have that selective sweep. Well, what we know is that many of the genes that show evidence of this sort of purifying selection or selective sweep are related to nutrition, metabolism, or immunity. So that gives us good evidence, in fact, that nutrition and immunity have played an active role in giving us a lot of the variants that we have today. But what's important to remember that it's not a one-to-one correspondence. That is, the one gene in the human genome that shows the highest evidence for this type of selection is the lactose tolerance gene, where there was a mutation in the promoter of the lactase gene that enabled it to be expressed through adulthood, and therefore allowed a source of nourishment, milk, that wasn't accessible to the rest of the population. But if you look at that polymorphism and you look at a heat map of where it is around the world, I'm sorry, this is the polymorphism, this is the phenotype of lactose tolerance, you see they overlap fairly well, but they're not identical. That is, no one gene variant, even the one that has the strongest signature of selection, no one gene variant completely determines what the phenotypic expression is going to be. We also see evidence here for a role of copy number variants in evolution. Here we have the amylase gene. If you look across human populations, you have between 1 and 9 copies of that gene. Individuals who have high copy numbers of that gene are from civilizations that converted to an agrarian type lifestyle, whereas the hunter-gatherers you find have the lower copy numbers. So you see these sorts of adaptations that have happened throughout history, driven by the food supply. And so these are other types of genes that have been shown to have these signatures of purifying selection, many of them related, again, to nutrition. What's interesting here is that the gene that enables lactose metabolism in adulthood is linked to one that enabled calcium transport. That is, after this mutation arose in the population and expanded that enabled milk consumption, the calcium then transporter then mutated along some sort of a temporal sequence. So what this tells us is that the idea that nutrition and diet are linked and that there are differences in nutrient or how nutrients are metabolized and how that the relationship between nutrients and health has a strong biological premise and background in evolutionary biology. But what we're not so sure about is what is the strength of the effect, or what we refer to as the penetrance, when you consider again that these are complex traits and genetics is only one factor that interacts with many other factors in determining what that connection between nutrition, food, and health is. So that leads to the question, does it really matter in public health? And I'd like to show you one example where, in fact, it has mattered. So all of you have probably heard the story of the MTHFR polymorphism. This is a fairly common variant that you find in human populations, where probably 80% of the alleles in this room have a C here with an alanine in the protein. Another 20% have a T with a valine in that spot of the protein. And this form of the enzyme is less stable and less active. If you carry that variant, you will have lower folate status, all things being equal, a higher folate requirement, risk for birth defects and miscarriage. But if you survive into adulthood here and you look at your colon cancer risk, and this has been repeated in several cohorts, what you see is if you carry the variant, your risk of colon cancer is reduced 70 to 80% if you maintain adequate in your folate status. So in fact, this is a very powerful effect from a single nucleotide polymorphism. You also see here that the allelic frequency varies across the globe. It obviously arose after the migration out of Africa, but it doesn't show a signature of selection, so it either accumulated through drift or it came from another archaic human. We don't know yet. But we also have good evidence from controlled feeding trials that that variant can affect the DRI for folate. This was a controlled feeding trial that was done by Marie Cornell, where she fed humans the DRI for folate, and these were, again, Mexican men. You can see here that during the feeding trial, their folate levels in their blood, serum folate levels, dropped because they were consuming more than the RDA. But after the 12 weeks, you could distinguish the CCs from the TTs. If you look at homocysteine, which is a functional biomarker of folate metabolism, what you see is for the CCs, the 400 micrograms was adequate as it didn't change their metabolism. But in the TTs, their homocysteine levels rose markedly. So in fact, this is an example of one single polymorphism that definitely affects nutrient requirement. In fact, the RDA probably is not adequate for Mexican Americans who harbor the TT genotype. Has this affected policy? And the answer is yes. So in 2015, the WHO published this guideline for optimal serum and red blood cell folate levels for the prevention of birth defects. Member states were coming to the WHO and saying, how much folate should we put in the food supply? And so they were asked to give guidance on this. So they put together a committee. And what is remarkable, I think, about this committee report is, number one, it's really the first time we used sort of a chronic disease endpoint, if you will, for nutrition guidance. But it also used big data integration in reaching that conclusion. That is, there were very little data and low quality data that linked folic acid intake to neural tube defect risk. But there were data that linked folic acid intake to red blood cell folate concentration, how the genotype modified that effect, and then how folate concentration affected neural tube defect risk. So they integrated data around a genotype model, what was the genotype distribution, that fed into a concentration model of how much folate they consumed and what was the effect of the polymorphism on blood folate levels, into a risk model of how that affected neural tube defect outcomes, and then through Bayesian updating, arrived at this completely computer-generated dose-response curve. So this is computer-generated, so it is computed evidence. Yet, the one study that did exist, which was a small study in an Irish population, these data points here aligned from that small study right along the big data curve. So this is really evidence of where a single polymorphism was used to generate a guideline on what the optimal level of folate should be to prevent neural tube defects, and is also an example of how you can use big data with small studies to confirm the big data and the generated evidence and confirmation that gives you a really high level of confidence in your recommendations. So a little bit about what will consumers need to know. So again, consumers will have available to them their genetic evidence. People are using all sorts of ancestry type companies that increasingly are giving health information. They had pulled back from that, but you're seeing again that health information based on your genetics is being distributed. But what will they really need to know in terms of how they use this information? So some have suggested, well, we need to classify subgroups for diets, and we've heard about that. And in fact, in the One National Committee report that was just released here on redesigning the process for the dietary guidelines for Americans, again, that report emphasizes right here that what we really want to know is how the DGAs can better prevent chronic disease, again, that chronic disease focus, but also take into account the range of individual factors that we've talked about. Diet is a complex trait in terms of its relationship with health. And what I want to point out again, there's a lot of data around studies around various types of diets and how they affect health in human populations, but a lot of this is observational data, and very little of it is long-term. That is, you conduct this experiment long enough that you can see the onset of chronic disease. But I want to point out this one study that's still not out yet. The pre-pub is out. That was done by David Threadgill and included people like Andy Feinberg on this paper. But what they did is they did a long-term study in mice where they took different inbred strains of mice and they fed them one of four diets, either the American diet, a Mediterranean diet, a ketogenic diet or a Maasai diet, and a Japanese diet. And they fed this to the mice for their entire life course. And they did full physiological profiling from how their epigenetics decayed over their lifetime to their whole metabolomic profile, and then even certain metabolic markers that we associated with health. And what you can see is that the best diet to maintain the health of this mice varied depending on what outcome you are interested in and what the genetic background was. So for instance, if you look at HDL cholesterol here, and you look at the Mediterranean diet here for this inbred strain of mice, it was very healthy, if you will, but not for the NOD mice, et cetera, et cetera. So again, one of the questions here is, what biomarker do you care about? What outcome do you care about? And depending on what outcome you care about, the genetics will point to a different diet to maintain long-term health. So this is really good biological premise, in fact, that diets should be matched to genotype. Of course, the challenge is how are we going to classify people? Because we can't classify them based on, inbred strain, if you will. So the next question is, well, what about classification of subgroups for nutrients? And this is based on the precision medicine model, where you have a prognostic test related to risk for some disease, some chronic disease, and then you have some predictive one to classify individuals. You have a predictive test of whether one drug A or B will work, and then you go and dose the drug based on that individual profile. And this has been somewhat, purported to be somewhat of a model for precision nutrition, although this has been problematic, especially in the diabetes world, where it is just so multifactorial in terms of the genetic contribution, the contribution of genetics and environment, that you end up really with a lot of difficulty classifying individuals, because there are so many variables related to most nutrition-related chronic disease. And does it really make sense to split the pie up in so many pieces that really constitute what a complex trait is? But this may not matter because, in fact, classification may not be necessary because of the whole trend towards real-time personal readouts. That is, people are collecting a tremendous amount of data on themselves. And there's apps for how many steps you've taken and your blood pressure and this and that. But now we're getting high-resolution data that's going to be available to most everyone. And the question is, what guidance are we going to give individuals when they find out all this information about themselves? And can we apply system network biology to this question? So this is just one example, and this does come out of Cornell. Some of my colleagues in engineering and nutrition have developed this Nutrifone, which is a microfluidic device, much like a pregnancy test, but it enables real-time readout of nutrient status biomarkers. They're up to 8 or 10 now. Functional biomarkers of nutrients, chronic disease markers, infectious disease markers. These are multiplexed, so with a little strip that the price will come down to a few dollars, you can get high resolution on your chronic disease status, infectious disease status, and what your nutritional status is. Then the question is, what do you do with that information? How do you match those up? And what is your cell phone going to tell you or not? Or are you going to go into WebMD? And I think what we've tried to do in my group to try to understand how best to help the consumer deal with the sorts of data that's coming down the pike is we've been working with the Microsoft Research University in Toronto to develop a stochastic model that really tries to understand network outputs in terms of folate metabolism. So using differential equations, we have recreated the entire folate network, but then overlaid onto that a simulation. And this simulation will run for hours or days if it has to until the system comes into equilibrium. But we have fed into this model all of the enzymes in the one-carbon network, including their compartmentation, all of their kinetic values. From the databases, all the known expression ranges of all the enzymes, the ranges for all nutrient inputs that go into that system. And then we've developed system-level biomarkers as readouts. So we can look at endpoints of the entire one-carbon system, which usually relate to genome-type readouts. So now we've tested this, we've published it, we've now expanded it, but now we can do sensitivity analysis for all the changes in the enzyme levels. So we understand what gene expression changes matter, which ones don't, and which ones reflect certain disease states or health states, which ones do not. And then once we have all of this different type of architecture of the network under different conditions, we can look at the dynamic range of inputs or nutrients, the 5 or 6 that are inputs into this pathway, required under different network states to maintain health. And I think these are the type of algorithms that are going to be needed so that when one gets all of that data from the cell phone, it can then plug into some sort of a model, a computational model, that will then be able to give them guidance on how they can optimize the function of their networks, given all of the data that they've collected. And so last, I just want to advertise that we are actually organizing right now a conference with some of my collaborators at UC San Diego and their engineering department in Germany at Cornell, which will be the first international conference on precision nutrition and metabolism. And what this really hopes to do, this conference, is bring together people in nutrition with engineers, with computer scientists, to really understand how we have a systems level understanding of nutrition to then give better guidance to the consumer who's going to have all of this data available to them. So thank you very much.